Hey, hello all. My name is Palash. Welcome to my first series of video with you Annex1 draft. Majority of this discussion is around sterility assurance assessment. I only consider those points where majorly this document is talking regarding the sterility assurance, and you have seen that it's more than 200 times, 250 times the guidance is talking about, sterility assessment and sterilization. So, a couple of critical aspect. It's a series one. So please be with me. I will be coming with couple of series to cover all entire guidance. It's quite a big document and cannot be discussed in a single session. So before going for my next slide or main presentation, I would like to request you all to subscribe my channel PRES. Pharmaceutical Resource and Education Services is a completely free site, and all the resources are available free of cost. So please follow my channel, press the bell icon, and get all the informations routinely. Thank you. So let's start from the introductions. Process, equipment, facility, manufacturing activities should to be managed in according to QRM principle. To provide a proactive means to identify, scientifically evaluating and controlling potential risks to quality. So it's very important. So you should look into all the aspect of your product manufacturing process, equipment, facility. So whosoever can have longer impact on the product sterility. So that is very important to assess through a QRM process. So whatever. So anyhow, the alternate approach can be used. Whatever the approaches mentioned in the guidance, you can use the alternate approach, but that's supposed to be supported by a good rationals and risk management. Ultimately the intent of this annex should not to be altered, right? QRM priorities should to be include good design of the facility, equipment, and process in the first instance. So it's not that you'll be looking for some loopholes and you'll be fixing after getting something done. Make the design robust, so the chances of failure in future will be minimized. So then implement a well designed procedure, so how we can drive your process and monitor your system. So it is very important. Design, you make a robust process, how to run your process, and then monitor for any kind of deflections you're finding in your process. So that demonstrate your design and process have been correctly implemented and continue to perform with the line of expectations. So it's very required to set up the initial expectations whenever you are making a new manufacturing facility, and whenever you're starting with the manufacturing in your new facilities. Exclusively monitor and test does not gives assurance of sterility. Just remember, making a exclusively or extensive testing cannot assure the sterility of your product, right? We'll come into this point later on as well, but keep in mind testing cannot suffice the issue of sterility, right? Manufacturer should take all steps and precautions and necessary to assure the sterility of the product manufactured within the facility, right? So what I'm talking about in my last slide as well. Sole reliance of the sterility or the other quality aspects should not place on the terminal process or the finished product testing, right? So it does not matter your process is terminal and you're going to kill all the microorganism end of your sterilization process, or you have a very extensive testing process from each and individual phase of your manufacturing or filling. So that does not make sense. You cannot able to demonstrate a good design or good control through this process. So starting with the pharmaceutical quality system, so it's talking about all non-conformance, right? All non-conformities such as sterility failure, inventor monitoring excursions, or deviation from the established procedure should to be investigated. The investigation should determine the potential impact on the process, product quality, and whether any other processes or batches are potentially impacted. The reason for including or excluding product or batch from the scope of the investigation should to be clearly justified and recorded. Hope this is a very precise one. I I need not required to repeat it. It's very clear. The instructions whenever you investigate, you need to follow all those critical steps. For equipment, whether any unplanned maintenance, you got breakdown, so we call it unplanned maintenance of the equipment critical to sterility of the product is to be carried out. An assessment of the potential impact of the sterility of the product should to be performed and recorded. So it is not appreciable, but the occasional breakdown whenever you are performing unplanned maintenance and during a process, it will have the longer impact on the sterility as well. So you need to assess. You should have a risk management, or you should have a clear cut instruction how to handle and whether any extend the risks to your the sterility of the product that need to be understood. One of the critical aspect I just want to mention in my first session, that personnel, which is very critical one, because most of the process required manual interventions, and whenever we are anticipating manual intervention, it's a chances of higher, um, uh, the chances of sterility assurance or chances of sterility failure is high because of those manual interventions. So only the minimum number of personnel, qualified personnel or approved personnel, so those who have challenged during the process and they are trained, they are expert. So those minimum number of persons required to present in the clean room. The maximum number of operator in the clean room should to be determined, documented, and validated during the activities such as initial qualification and aseptic process simulations. Right. So, as not to compromise the sterility assurance, and this is particularly important during the aseptic processing. So, well, as I'm talking about, the personnel should to be well trained, person should to be, the practice should to be, observe, monitor, and finally that required certain kind of qualifications whenever we are going to include those personnel for any kind of aseptic process handling. So it's required to undergo through a media field where you will be qualify that person and his activity. So majorly challenge with the maximum person in your uh, facilities, and try to use as much less during your routine process. So that is the way we handle the personal qualifications. Friend, thank you very much. So this is a small first session. So soon we'll be coming with the second session of sterility assurance considering EU Annex 1. We'll be happy to get feedback from the first sessions. So do you like it? Any questions or anything you want to include or any clarification you're looking for? Please feel free to get back to us and write in the comment section of this video. Thank you very much. See you soon.